So we have here some starch, vegetable oil and sugar solution, and we also have egg albumin, and we're going to be testing these for the presence of simple sugars, starch, lipids and proteins. To do that, we're going to be using the following reagents: iodine solution, biuret reagent, ethanol and benedict reagent. I'm going to start by adding egg albumin to the test tube furthest to the left. Then I'm going to add some sugar solution to the next test tube. Then I'm going to add some starch. And finally, some vegetable oil. And then the first test we're going to do is the test for starch. So we're going to add iodine to each one. So the first one we're going to add iodine to is the egg albumin. As you can see, there's no change in color, so that's negative. Next one is the sugar solution. Once again, it stays orange-brown, so that's negative. Next one is the starch solution. That changes to blue-black, so that's positive. And the last one is the vegetable oil. That stays orange-brown, so that's negative. Now I've got the same for foods, and I'm going to add biuret reagent, and this is the test for protein. First one I'm going to add it to is egg albumin, and as you can see, the color changes to purple, which indicates that protein is present. Next one is the sugar solution. No change in color, so that's negative. Next one is starch. No change in color, so negative. And next one is vegetable oil. Stays blue, so no change in color, so that's negative. So the only one that's positive is the egg albumin. And you can see the comparison between the egg albumin and the vegetable oil there. The egg albumin is clearly purple, so that is a positive result indicating the presence of proteins. I've set up our 4 foods again, and the next one we're going to use is ethanol to test for the presence of lipids. All 4 of these already have water in them. If they didn't, we would have to add water to it. The first one I'm going to add ethanol to is the egg albumin. And as you can see, a cloudy white layer has been formed there, which indicates the presence of lipids. If I add a bit more ethanol, you can see it more clearly. There is a cloudy white layer there at the top. The next one we're going to add ethanol to is the sugar solution. And as you can see, there's no cloudy white layer there, so that's negative. Next one is starch. There's no change, there's no cloudy white layer, so that's negative. And the next one is vegetable oil. And you can see a layer has formed there, which is positive for lipids. I've set up the 4 foods again, and this time we're going to be using Benedict's reagent to test for the presence of simple sugars. With Benedict's reagent, we need to heat these for 10 minutes. So I've set up a water bath set to about 95 degrees Celsius, and we're going to put the food samples with Benedict's reagent in the hot water for about 10 minutes. So the results with the Benedict's reagent, you can see that the egg white changed the color of the Benedict's reagent, so there's some reducing sugars in egg white. The sugar solution turned yellow, and there was no change with the starch and the vegetable oil. They stayed blue, so those are both negative.